So, hello everybody, dear attendees. So we are very pleased to welcome you today for our third webinar on cyclodextrins, organized together with Roquette and CarboHyde, and today with Aquilon Pharma. So the first webinar held in June 2023 and was dealing with cyclodextrin in biopharma. The second webinar took place in October 23 and discussed about cyclodextrins in pharmaceutical applications from manufacturing, properties, safety, and regulatory aspects, as well as the next generation of cyclodextrin as therapeutics. Today, we will deep dive in cyclodextrin formulations from oral, parenteral, transdermal to inhalation dosage forms. For that, 4 presentations will be given to you today. Each presentation will last about 20 minutes with 5 minutes for question at the end. After the presentation session, we will go into a panel session with our guests, Professor Paul Maes, Chief Executive Officer of Aquilon Pharma, and 3 Roquette experts in cyclodextrins, Julien Parc, Damien Truffin, and Hélène Blouet. So the topic of today, cyclodextrin formulations, a journey from the benchtop to the patients. During the presentation session, do not hesitate to type your questions in Q&A section, and our presenters will respond to your question after the presentation or during the panel session. We will start now with a first presentation titled Preparation of Parenteral and Oral Cyclodextrin-Based Formulations and Their Physical Characterization. For this presentation, I'm glad to welcome Dr. Carmen Popescu. Carmen is Global Technical Developer at Roquette with an extensive knowledge on pharmaceutical technologies and a particular liking for cyclodextrins. So, Carmen, please, the floor is yours.
Hi, everybody. Today I have the great honor and pleasure to share with you a few practical aspects of solubilization of BCS class 2 and 4 drugs with cyclodextrins in parenteral and oral formulations. Generally speaking, when we have a drug candidate and we couldn't solubilize it by salt formation, pH adjustment, or using co-solvents, and this drug has a high log P and a high melting point, and it is going to be administered IV at high dose or orally at low dose, and the drug has a suitable molecular shape, we can say, just based on physical properties of the API, this drug is a good candidate for cyclodextrin complexation. In this slide, there are highlighted few but important characteristics of an API, and I call this birth certificate of the API because when I'm talking BCS class 2 and 4 drugs, we'll see that they have some common characteristics like low intrinsic solubility, low water solubility, log P is high, high melting point, and so on. So we have to understand very well all these physical properties in order to know how to handle their solubilization. What are cyclodextrins? They are cyclic oligosaccharides derived from starch. They have a bucket-like structure with an inner hydrophobic cavity and a hydrophilic exterior. In nature, we find alpha, beta, and gamma cyclodextrins. This unique structure allows them to form inclusion complexes with hydrophobic compounds by non-covalent bonding within the inner cavity. In this presentation, I'll focus on solubilization of BCS class 2 and 4 drugs with native beta-cyclodextrin and modified beta-cyclodextrin. As you can see in this table, intrinsic water solubility of native beta-cyclodextrin is 185. That is a limiting factor when we are looking to have higher degree of solubilization of BCS class 2 and 4 APIs. By derivatization of native beta-cyclodextrin, by hydroxypropylation, methylation, or sulfobutyl ether sodium salt creation, the native cyclodextrin limitation is overcome because these derivatives, as you can see in the table, have higher solubility, more than 50% for hydroxypropyl cyclodextrin and sulfobutyl ether cyclodextrin, and 20% for methylated cyclodextrin. As you can see in this slide, on the market, you can find APIs solubilized with cyclodextrins in different type of forms, from liquids to solid dosage form to topical and oral disintegrating films and tablets and all kind of dosage form. In order to successfully solubilize a drug with cyclodextrin, we have to know a few details like the number and type of atoms on the drug molecule skeleton. Also, the molecule shouldn't have more than 5 condensed rings. The more insoluble a drug, better candidate for cyclodextrin complexation. Melting point has to be under 250. The molecular weight, generally speaking, between 100 and 400, but we successfully solubilized 600 and 1000 molecular weight. Electrostatic charge, it is not a problem for solubilization with cyclodextrin, but pKa is critical because we have to know that the drug has a pKa, because we have to do solubilization in a pH range where the drug is totally insoluble. Also, we have to know if the drug has some chemical and photostability issues. How the inclusion complexes are formed. Situation is like this. We are talking about an API which is hydrophobic, meaning water repellent, doesn't like to stay in water, is not happy in water. When this molecule is closer to a cyclodextrin molecule, which has a hydrophobic inner cavity, gladly the drug will go inside in central cavity, which is hydrophobic, because there the molecule will be happy. And in this way, we form a complex. But this complex formation, it is thermodynamically driven. It is not driven by covalent forces. Anytime the drug inside of cyclodextrin complex is in equilibrium with the free molecule in solutions. Anytime if the complex between the drug and cyclodextrin it is not strong enough, meaning the non-covalent forces, non-covalent bonds created inside of cyclodextrin cavity are not strong enough, the drug molecule can come out in solution. The driving forces of complex formation are electrostatic interaction, van der Waals interaction, hydrophobic interaction, hydrogen bonds, and many times the conformational structure of the molecule can help for the molecule, the hydrophobic part of the molecule to go inside of cyclodextrin. But before complex formation, these forces have to be stronger and enough to release the rich enthalpy water molecule which are in the inner cavity of cyclodextrin. What type of complexes can be made between a drug and a cyclodextrin? Can be one drug molecule, one cyclodextrin molecule, can be one drug molecule and two cyclodextrin molecules, or if the drug molecule is small, can be two drug molecules inside of one drug cyclodextrin molecule. So let's see how cyclodextrin can encapsulate a drug. In this video clip, you'll see we have one beaker with distilled water, one beaker with cyclodextrin, and two beakers with phenolphthalein in alkaline solution. They will take for the pH in both beakers, distilled water and cyclodextrin, we have the same pH. Then they will drop by drop add phenolphthalein in each beaker. And as you can see, in distilled water, the color is accumulating, while in beta-cyclodextrin beaker, there is no color. So it is sort of magic event here. What is happening? At the end, you see the color in distilled water became more intense, and no color in phenolphthalein will take the pH, and this is done. Let's have a look what is happening with phenolphthalein. Why phenolphthalein became pink? This is just chemistry, so I would let you watch the video, the simulation. So that is the phenolphthalein molecule in a simulation stage. Let's have a look about our cyclodextrins. So there are different models to simulate cyclodextrin structure, and you have stick model or surface model where it looks like a donut. Then you have stick and surface model together, showing you the distribution of the atoms within cyclodextrin. And then the most representative is this picture showing all the forces, especially the electrostatic potential on the surface of cyclodextrin. Now let's see what is happening during our experiment. Phenolphthalein poured in the cyclodextrin solution. So you can see the phenolphthalein is getting inside of cyclodextrin, is getting inside in those available molecule of cyclodextrin, and it is not a chemical reaction, as I mentioned, it's just a thermodynamic interaction. The closer to an empty cyclodextrin, better chance for phenolphthalein to enter within the cavity of hydrophobic cavity of cyclodextrin. Now let's see if we look to one molecule of phenolphthalein meeting a molecule of cyclodextrin. So we have phenolphthalein with the rearrangement of the atoms, electrons, in fact, in the atom, becoming a pink color. And now it's going to meet a cyclodextrin molecule. And before meeting the cyclodextrin is rearranging, that meaning the steric fitting inside of cyclodextrin. So what we have to see, and is very important, when we say a cyclodextrin molecule of API will go inside of cyclodextrin, that doesn't mean all molecule will go, just those hydrophobic parts of the molecule of API. So here you have the real interaction between the cyclodextrin hydrophobic inner cavity and the molecule of phenolphthalein. So again, we have to keep in mind that not all molecule of gas will go inside of a host cyclodextrin molecule. So that is very important. That's in order to create liquid or solid dosage form, we have to run so-called phase solubility study by screening the available cyclodextrins and evaluate by HPLC the phase solubility. Also, next step will be, if we want to do solid dosage form, to create solid dispersion of cyclodextrin complexation by different methods: spray drying, kneading, freeze drying, physical mixture, screening the cyclodextrins, and then based on differential scanning calorimetry, we can prove the complex formation in this solid dispersion, and the dissolution profile will allow us to select the process and the best cyclodextrin to solubilize the drug in a solid dispersion. API cyclodextrin complexes can be classified based on Higuchi and Connor phase solubility. What is phase solubility is evaluation of solubility increase of API with increased concentration of cyclodextrin at supersaturation, and it is expressed in molar units. We can get an AL profile, which is a linear regression, and many times we can get an AP profile with a positive deviation from the AL linearity, and that can express a different stoichiometry, meaning one cyclodextrin can complex 1, 2, or 3 API molecules, or in many situations there is no inclusion complexes. Another situation is a AN deviation, a negative deviation from linearity, and in this case, one drug molecule can complex with 1 or 2 or 3 cyclodextrin molecules. There is a limited solubilization profile, B profile, and that is mainly attributed to native cyclodextrins. The phase solubility curve profile do not certify the formation of inclusion complexes. They only describe how increasing cyclodextrin concentration influence the drug solubility. And that is because we know that cyclodextrin can form non-inclusion complexes or complex aggregates like micelles, and those are not inclusion complexes. Let's take into consideration few case studies. Let's start with Zaplon, which is a BCS class 2 drug with very low oral bioavailability due to its low solubility. How can we run phase solubility? Briefly, based on Higuchi and Connor, we have vials in a certain range of concentration for each cyclodextrin. Then we add to these cyclodextrin concentrations excess amount of API. We mix the vials at room temperature for 1 or 2, 3, 7 days until we reach supersaturation in the vials. Then aliquots of the solution are filtered through 0.22 micron syringe filters and analyzed by HPLC. And then the concentration of solubilized drug in molar or millimolar units are plotted for each cyclodextrin concentration expressed in millimolar or molar, and this represents the curve for phase solubility of the drug in respective cyclodextrin. Phase solubility curves are representing linear regressions, so the conclusion is Cyclodextrin is forming 1-to-1 complex with each cyclodextrin. These experimental curves are helping us, based on the slope of the curve and the water solubility of Cyclodextrin, to calculate, as per equation 1 and 2, the stability constant and complexation efficiency. Looking into the table, we can see that the slope for methylated cyclodextrin, CRISMAB, it is closer to 1. That means you can solubilize almost 1 mole of the drug in 1 mole of cyclodextrin. For the other cyclodextrins, the efficiency it is lower. So, phase solubility is absolutely necessary to be run in order to be able to calculate the stability constant and complexation efficiency. As I mentioned before, just solubility increase, it is not a proof for complex formation. In order to prove complex formation, we have to run a method like differential scanning calorimetry to show that the exothermic peak, which is specific for the API, is disappearing when we have drug and cyclodextrin complexes. Also, if we look in the table, let's say for 50 millimolar of each cyclodextrin, we can see how many times we increase the solubility. In the case of hydroxypropyl beta cyclodextrin, 5.6 times. In the case of methylated cyclodextrin, 9 times. In the case of sulfobutyl ether cyclodextrin, 3 times. This increase of solubility of API is allowing us to rank the efficiency of each cyclodextrin after we proved that is due to complex formation. Up to now, we know that phase solubility is allowing us to evaluate the best cyclodextrin for our formulation based on complexation efficiency and the stability constant. Then differential scanning calorimetry is helping us to prove that we have complex formation. The next step is to show that these liquid formulations are stable at 25 and 40 degrees Celsius. And then, based on the stability evaluation, we can rank which one of these cyclodextrin is really the best. If none of them is helping us to keep the drug in liquid phase, the solution is to lyophilize after solubilization so we can create a reconstitutable powder for injection. Moving from liquid state to solid state, let's see what do we need to create solid dispersion inclusion complexes. The methods used for creation of solid dispersion, as you know, are spray drying, freeze drying, knitting, to name few of them. But for all this, the common denominator is this. we have to find a volatile hydrophilic organic solvent in which our API is soluble. Then we have to find the miscibility ratio of this volatile hydrophilic organic solvent with water. This you can find in some catalogs. Then we prepare a saturated solution of the API in the organic solvent, and we already know what is the ratio needed based on the phase solubility of the cyclodextrin, which will be dissolved in water. Then we'll mix the solution, as I mentioned, solution of API with the solution of cyclodextrin in the miscible ratio, and at the end we have to get a solution. If we have a minimal turbidity, that means we don't have the right miscibility ratio between these two solutions, and the drug will crash out. In the case of Zaleplon solid dispersion inclusion complexes by spray drying, based on the phase solubility, we know the ratio between cyclodextrin and API. In the case of hydroxypropyl beta-cyclodextrin with methanol as volatile organic solvent, and for Kreismeb we use acetone. And of course, you establish the spray drying parameters, and then at the end you are going to get a spray dry solid dispersion with a certain content of API, which will be evaluated by HPLC. In creating Zaleplon solid dispersion by freeze drying, we use the same combination of organic solvents, volatile organic solvents, and cyclodextrins. We did the freeze drying, and then we got a solid dispersion powder, which has been evaluated by HPLC. We also created the dispersion inclusion complexity by physical mixture using a V-blender or a turbula mixer, and mixing the API and cyclodextrin in the ratio as per phase solubility. And of course, this kind of solid dispersion is less efficient than the spray drying, lyophilization, or knitting, but it's always good to have sort of reference. Next step was to evaluate the complexation efficiency for each type of process by evaluating how much powder contains, let's say, 10 milligrams of drug. The next step is to decide which one of these solid dispersion methods is the best, and that can be done through differential scattering calorimetry and dissolution profile assessment. Using differential scanning calorimetry, we can evaluate which one of these processes is the best by measuring the disappearance of exothermic tip of API in the solid dispersion associated with certain solid dispersion method. And the next step was to evaluate the dissolution profile. As you can see, in the case of Kreismeb, spray drying was better than lyophilization and better than physical mixture. But surprisingly, when we look to hydroxypropyl beta-cyclodextrin dissolution profile, we found out that spray drying is better than physical mix and better than lyophilization. That's why always we have to compare the cyclodextrin and the method used to create the solid dispersion in order to pick the right one. From differential scanning calorimetry data, we can see that spray drying Zaleplon in Kreismeb, the crystallinity, it is 0. That means we have a pure amorphous powder. From dissolution evaluation parameter, for each cyclodextrin and process, we can evaluate the cumulative drug release, 5 minutes, 30 minutes, dissolution efficiency, and other parameters. Let's have a look on another API, zotipine, which due to low solubility has a very low oral bioavailability. I selected this case study and this slide with zotipine to highlight 2 points. One is when you run phase solubility, you have to run with all available cyclodextrins in the lab because you never can predict what is going to be the best choice. In this case, the native beta-cyclodextrin is giving a better complexation efficiency with zotipine than the derivatives. Also, with small amount of native beta-cyclodextrin, just 60 millimolar, you can increase the solubility of zotipine for 870 times. And this is the best way to prove nothing can be predicted a priori. You have to run the phase solubility and then draw the conclusion. Also, looking to the solid dispersions with different processes and different cyclodextrins, surprisingly, when you look to CRISMAB dissolution profile of zotipine, physical mix is better than spray drying. That was really, really surprising for me. So again, when we talk cyclodextrins, nothing can be predicted. We have to run the experiment thoroughly and then to draw the right conclusion. In this slide, you have a list of the drug solubilization studies in different type of formulation, liquid or solid dosage form, available to you upon request. Next video clip will visualize through molecular dynamic simulation at microsecond timescale how API is released from the complex at the interaction with phospholipid membrane bilayer. At the end, thank you very much for your attention. And if you have any question, contact me at your convenience.
Thank you very much, Carmen, for this very interesting presentation. We will not have time for questions now, but we will take them in the panel session. Thank you very much again. To continue, the second presentation is titled Cyclodextrins in Liquid, Transdermal, and Other Non-Traditional Forms. For this presentation, I'm glad to welcome Dr. Balázs Kondoros. Balázs first encountered the topic of cyclodextrin in his undergraduate pharmacy study. He later started to do experimental work on this topic, from which he obtained a PhD degree in this year, in 2024. He is most interested in areas where cyclodextrin are used as excipients in unexpected and innovative ways. And currently, Balázs is working as a formulation scientist at CarboHyde. So, Balázs, the floor is yours.
Hello and welcome everyone. I'm Balázs Kondoros. I'm a formulation scientist at CarboHyde, and today is my task and my pleasure to speak about cyclodextrins in non-traditional dosage forms. So, in the ever-evolving pharmaceutical industry, it is always a great challenge and a good potential to deliver APIs in new dosage forms for new applications and new administration routes, and also it applies for cyclodextrins. So let's see what is considered non-traditional in the world of cyclodextrins. We always say that they are used as excipients, but in a non-traditional way, they can be used also as APIs. In the terms of application, they are used for solubility and stability, enhancing and also taste masking in a traditional way, but new applications considered as vaccine adjuvant, cryoprotectant, drug delivery systems for RNA and DNA, and the list goes on, of course. Yeah, and for administration routes, traditional ways are IV solution and oral solids, and we can say that practically everything else is non-traditional. But this is something that not defined, and something that is not my task today to define, because you can say that, okay, but we know that for decades that they can be used as APIs that are already on the market with vaccine adjuvant properties. So what is considered non-traditional administration route. So this is not my task today to define that, but I will give an outlook and some very interesting examples for the non-traditional applications. Something I would like to highlight is that something is traditional is not equals to outdated, and something non-traditional is not novel. Something can be non-traditional in these terms and can be something nonsense at the same time, and something can be very well patented, something can be very new and in a traditional applications or traditional administration route. So as I said, they are multifunctional excipients. They are used for decades. There are already more than 100 pharmaceutical approved products. And I would like to show that very handy table produced by the European Medicinal Agency with different administration routes, and in those applications, what are the already approved cyclodextrins in different products. In terms of trends, we can say that mostly parenteral and oral patents are appearing, but the ocular and nasal and also pulmonary delivery is very promising, but there are neglected areas like topical and transdermal areas, and we saw a very fast growth in the use of cyclodextrins in the early 2000s. And it is not surprising, because you can practically you can say it for every pharmaceutical areas. These are the trends, and as you can see in that table, in the last 5 years, the products that are appearing on the market are usually IV solutions or lyophilized powders for solution. I will highlight the nasal powder with glucagon produced by beta-cyclodextrin complex, but I will tell you more about it later. So again, with the question mark, these are non-traditional dosage forms. So there are already on the market with, for example, oromucosal spray with a known API, with a known cyclodextrin complex, but with a twist, with something new. Also, there are chewing tablets with beta-cyclodextrin, and we already know these applications, but we can introduce them to the market with something new. So the first topic I would like to speak is the ophthalmic products with cyclodextrins, and why we are using cyclodextrin for ophthalmic. Of course, they are improving solubility, permeability, and stability of the drug. That's something we already know. And something very useful is the reducing local irritation effect of cyclodextrins, which can be very handy in an eye drop, for example. And all in all, they are leading to improved drug efficacy. In the table, you can see that there are plenty on the markets, not just eye drops, but some ointments also. They are using beta and gamma cyclodextrin derivatives, and also native cyclodextrins in these formulations. But there are some issues. For example, when you are using anionic cyclodextrins, we really like to use sulfobutylated beta cyclodextrin, but it has the backside that it raises osmolality in a great way. So Savalent technology is something that overcomes this issue, where we are using the anionic cyclodextrin and the API as counterions, producing pharmaceutical salts, and by that you can reach up to 7:1 API-CD ratio, and by that you can decrease the osmolality, you can control the pH, and with this higher potency you can lower the volume, and these are all properties that are very, very welcomed in the world of ophthalmic product. And this concept can be also used for dual API products. Another problem is that to deliver some APIs to the posterior tissue of the eye, with conventional therapy, you can reach the back of the eye in a small ratio, and of course invasive drug administration is something you can do, but something what we really want to avoid if possible. And with the Oculis Optireach technology, it seems possible. It already passed phase 3 clinical trials, and it contains dexamethasone as API and gamma cyclodextrin, which has the disadvantage to form aggregations. We always say it's a disadvantage, but in this application, they used it as an advantage and deliver the API in a delayed administration, and by that you can reach the posterior tissues with this cyclodextrin formulation. Another interesting area is the electrospinning. You maybe already know that they are used as with polymers, and you can combine cyclodextrin with polymers. You can also use polymerized cyclodextrins without added polymers, and you can use only cyclodextrins for this application. And there is an interesting example here. I would show that using HPBCD for enhanced solubility, and it also enhanced the physical-chemical properties of the electrospan met and the spinability. And you can use this electrospan ophthalmic insert for eye delivery, and with the benefit of elimination of the need for preservatives, which also a great challenge in ophthalmic delivery, since cyclodextrin can encapsulate preservatives, and by that they are lowering the effects of that agent. The next step is the skin-related product, and the cyclodextrins are used for solubility and permeation enhancing. But I would like to highlight one example, which contains corticosteroids, where they used water-based ointments, where cyclodextrin increased the delivery of the API and delayed the drug release in oily-based vehicles. Both of them can be useful. You just have to pick the right formulation for your application. And in the table, you can see that there are already on the market, not all of them are pharmaceutical applications, but you can see that there are creams, ointments with beta and gamma cyclodextrins. And here I would really like to highlight that they are used in cosmetics, and they are very handy for solubilizing and stabilizing. You can avoid using co-solvents. It is easier to incorporate some materials, and there are process improvement properties of cyclodextrins, for example, using incompatible compounds in the same system, change viscosity. And in that little table, you can see that not only native cyclodextrin can be used, but also the derivatives of beta cyclodextrin. In transdermal delivery, there are, of course, some new applications, for example, microneedles, where cyclodextrins are useful for permeation enhancing by enhancing the solubility of the API. For example, in this example, they were using hydrogel forming microneedles, but these microneedles were not containing the cyclodextrin. They used the compressed tablet as a drug reservoir and put on the hydrogel, and by that the administration were enhanced. For nasal administration, the goal can be systemic, local, or nose-to-brain administration. Cyclodextrins are used in all for those goals in publications, and they are usually solubility and permeability enhancing agent in these. An interesting example is a parenteral hormone application where enhanced nasal delivery was approved by cyclodextrins, and also the drug aggregation was improved by the use of cyclodextrin. And there is already one product on the market which I already mentioned, the glucagon-containing beta-cyclodextrin formulation, which can be used in emergency situations for unit dose as a unit dose nasal powder. For pulmonary administration, there is nothing on the market yet with cyclodextrins, but there are great potentials. So the goal here can be also systemic or local, but something that is something that we should keep in mind, that the part of the innovation is also the use of the device, the engineered device, to deliver the cyclodextrin-based formulation. And there are a lot of publications containing cyclodextrin-based formulations with a variety of drugs for improving lung absorption, improving plasma concentration, and also the physical properties of these formulations are very very critical. So that is something that cyclodextrin can improve. It can be surprising, but in suppository products, there are more on the market with cyclodextrins, and they are very useful for decreasing the interaction with the base or inhibiting the reverse diffusion into the vehicle of the API, and of course the irritancy of the API. And as you can see, beta-cyclodextrin and beta-cyclodextrin derivatives are used in many cases in pharmaceutical products on the market. There is two examples in the patents that I would like to highlight, that cyclodextrin can increase the systemic effect and also the local effect if you choose the right administration and right formulation. In the first case, the pediatric cancer compound was delivered by the cyclodextrin as a solubilizer, but in the other case, for local retention, cyclodextrin was used with two different APIs. So as a summary, I would like to show you the list that is not complete. It is always an ongoing list, and some challenges that cyclodextrin are very useful, but they can change the physical chemical properties of the whole system. For example, using an ionic cyclodextrin can increase the osmolality. Using them can be cause incompatibility with other excipients, for example, preservative in eye drop formulations, and incompatibility with non-aqueous bases, which is something that we can overstep, but something that we are not used to because we are always considering aqueous bases. And also it is a great challenge because there are non-existing toxicity data of cyclodextrin derivative in specific administration route. So this is a burden to step on the market because we have to approve these cyclodextrin derivatives. And thank you for your attentions, and I'm here for the questions. Thank you.
Thank you very much, Balázs, for this very interesting talk. We are still a little late, so I propose that we take all the questions in the panel session as well. Thank you again. So now time has come to start the third presentation titled Hunting for Cyclodextrins, Analytical Assay Methods and Biological Fluids. For this presentation, I'm glad to welcome Tamas Soadja. Tamas is the Chief Executive Officer of CarboHyde and has more than 16 years of history with cyclodextrins. So Tamas, the floor is yours.
Thank you, Nicola. Can you confirm if you can see the slides moving?
We can. Yes. Perfect.
Okay, perfect. Thank you. So, hi, everyone. Now this session will be less about cyclodextrin formulations and much more about their analysis and especially analysis in biological fluids. And the reason why we picked this topic is that it's obviously a very a question that's coming up a lot of time, especially with cyclodextrins becoming therapeutic ingredients too. Yet there are no comprehensive reviews on the topic, and if you want to check the information, you have to pick from different sources. So this is a compilation of more or less what's available on the topic in the literature. First, I would like to talk a little bit about what are the challenges of cyclodextrin analysis in general, and then would like to show you how biological samples are analyzed, or how cyclodextrins in biological samples are analyzed to be more accurate. And there are three types of approaches for that. When the unmodified cyclodextrin itself is the target of the analysis, there are some approaches which use fluorescent labeling of the cyclodextrins and some that use radial labeling. So what are the challenges that we're facing when we would like to analyze cyclodextrins? Well, obviously, one of the biggest challenges is that many of the cyclodextrins that we use in formulations, either as excipient or as active ingredient, are not single compounds. So we are dealing with composites, with multi-component mixtures that are inherently more difficult to quantitate, more difficult to follow the pharmacokinetics, the pharmacodynamics, the accumulation, the metabolism, the degradation, and so on. Of course, in some cases, we're lucky with single isomers. Cyclodextrins have no UV absorption, so we have to rely on different analytical tools if we want to detect them. The functional groups that they have are highly similar, so separating them can pose a challenge. Sensitivity, as you'll see, will become a big issue. Although in many cases, cyclodextrins are administered in fairly high doses currently, but if we think about them evolving as really active therapeutic ingredients, probably the doses will get lower, and that will lead to the necessity to have sensitive analytical techniques. Of course, we'll have to get rid of the unwanted components of the matrix without compromising the cyclodextrins themselves, which can be similar to some matrix components because they're highly soluble and so on. So they are not typically drug-like molecules. And as we heard before from Balázs, they can also aggregate. Oh yeah, and one other aspect, of course, they can also form complexes, which can also complicate the analytical aspects. I'd like to quote a very famous cyclodextrin researcher here, Joseph Pita, who said once over 30 years ago that mixtures are beautiful if properly characterized. Well, with cyclodextrins, this is certainly a challenge. So still not in biological fluids in general, how cyclodextrins are analyzed when we are focusing on identifying them and quantifying them. So the identification tests in the pharmacopoeia for the cyclodextrins themselves are infrared spectroscopy, HPLC retention with different kind of detectors, refractive index, evaporative light scattering, or CAD. For the beta cyclodextrin itself, there's a reaction with iodine, and for the sulfobutyl cyclodextrin, since it's negative, there is a capillary electrophoretic method. For the assay testing, it's usually HPLC, or there is no assay testing at all, even in the pharmacopoeia. And to have the degree of the substitution, it's either NMR or capillary electrophoresis. Of course, on the top of that, there are cyclodextrins which have no compendial analytical tools. So in these cases, every user, every company, every producer can develop an analytical method of their own, like in the case of Sugammadex, which still have no monograph, despite the fact that there are many impurities to be detected here, as you can see. So how did the biological or the in vivo analysis of cyclodextrins start? Obviously, after they were discovered not to be toxic and there was an intention to use them in pharmaceutics, there was a need to understand their pharmacokinetic behavior as well as their safety profile. So in the 80s, more or less, these experiments were carried out and published. So some of the key features of their pharmacokinetics, they are technically no oral bioavailability, as you can see here. It's the highest is 3% orally. It's the HPBeta cyclodextrin among the approved ones. The absorption is only via passive diffusion. They're non-digestible apart from the gamma cyclodextrin excreted in urine. They're not metabolized significantly, and they disappear very rapidly from the systematic circulation within the frame of a few hours. So let's see how the analytical tools to capture cyclodextrins in these biological fluids developed. And now we're starting, as a reminder, with the methods that analyze the cyclodextrins as they are unlabeled cyclodextrins. So it started, we could say, with beta cyclodextrin, or that's the first example. As you can see, it was still in the late 80s, at least the publication was in the late 80s, and the chromatographic tools were used for the analytics. In the very first methods, the typical approach was to do a post-column complexation. Here, as you can see, they used phenolphthalein, and that was used for the detection, for a UV-based detection. A similar method was used and published in 2 separate articles for the beta cyclodextrin and the hydroxypropyl beta cyclodextrin. And as a representation, I'm showing you the hydroxypropyl beta cyclodextrin because it's more interesting. You can see that it's not a single peak. It comes in a very wide retention zone with a poor detection limit, and they used only the highest peak for the quantitation and the whole fingerprint for quality control. And even in the article, they proposed that size exclusion chromatography could be a suitable tool for a one-peak method, which was reality, as Satmari showed a few years later on HPBeta cyclodextrin in plasma and urine samples. So here they still used a post-column complexation, but they used size exclusion chromatography. And as you can see in the upper right corner, HPBeta cyclodextrin shows a very nice single peak. They could even improve the sensitivity of the method. The detection limit got lower. They had to use a more special sample preparation with solid phase extraction because of the inhomogeneous nature of the cyclodextrin. But as you can see, this was a method that was much more suitable. HPBeta cyclodextrin, it was linear and it was more or less accurate at that time. Now, another approach from the detection point of view was published in the mid-90s by Kubota and co-workers, where unparametric detection was used to determine beta cyclodextrin and glucosyl beta cyclodextrin in plasma and urine. And as you can see, the two types of cyclodextrins could be determined even in parallel, and the recovery of the dose in urine could be followed very nicely with the technique. Again, a bit later, in the late 90s, the research started to move from post-column for that. With the first one, you can see it's for the detection of methylated beta cyclodextrin, and the complexing agent they used was naphthol, which was not only to form a complex with the cyclodextrin, but also to act as a fluorophore, since fluorescent detection was used in this method with a nice limit of quantitation. And as you can see, very nice and sharp peaks, even in the presence of the active ingredient or without it. And it was still size exclusion chromatography that was used in plasma samples and cell lysates. A similar approach was used by Seman et al., but here the analyte was hydroxypropyl gamma cyclodextrin. As you can see, similarly, fluorescence detection was approved, and they showed that a wide variety of biological samples could be applied for the analysis, like rabbit plasma, humor, monkey plasma, monkey urine, and so on. And here you can see the samples. Obviously, at the limit of detection. So here is the detectability limit, which was in the nanomolar range. The peak shape is not the nicest, but it's still detectable. When cyclodextrins so when at the time where cyclodextrins were developed as active ingredients, obviously their huge efforts were directed into this field because in that case they moved from excipient status to active ingredient status, and the pharmacokinetics and the safety profile became much more important. And also the dose of Sugamadex, which is used in therapy, was much lower than the ones that were used before. So there was an urgent need to develop methods which are much more sensitive and much more accurate, that could be validated under GLP and then GMP, which was for the excipients at that time certainly not a necessity. So for Sugamadex, of course, this all started with the originator, who is now MSD. And for this particular compounds, two different liquid chromatography and MS-based, so LC-MS-MS-based detection assays were developed, and they were both validated. What's interesting is that you may recall that Sugamadex forms a very strong complex with rocuronium, and the methods cannot differentiate between rocuronium or between the uncomplexed and complex sugar gamma-dex, but they can selectively track it. And as you can see, the quantitation limit is over an order of magnitude lower than before, but still 2 orders of magnitudes higher than what was for rocuronium. And here you can see on the figure the analysis of different types of patient populations with different doses of sugar gamma-dex. The same was, of course, needed for hydroxypropyl beta cyclodextrin when the Niemann-Pick story started. And as a reminder, hydroxypropyl beta cyclodextrin, unlike sugar gamma-dex, is a multi-component mixture. So it was more challenging, but the principle was more or less the same. 2 types of LC-MSMS-based assays were developed, one using in-source fragmentation and the other one using reversed phase ultra-performance chromatography. And the analysis, what I'm going to show you, was done in human plasma and cerebrospinal fluid samples. These methods were validated, so these are actually the methods that were used for the clinical trials. Proteins were precipitated, and samples after dilution was just directly analyzed. Here in this figure B, you can see the first method, the in-source fragmentation method, and on figure C, you can see the reverse phase method and how nicely the cyclodextrin and the different species of the cyclodextrins were detected. Of course, there was a need to develop sugar gamma-dex, not just for HPBeta cyclodextrin, but for sulfobutyl cyclodextrin. The methods do not tremendously differ from those of the hydroxypropyl beta cyclodextrin. This was mostly necessary because questions about renal accumulation and toxicity. So the methods and the analytical results were focusing on patients who are under dialysis. But in principle, the methods that are used for the sulfobutyl are very similar to the ones that are used for hydroxypropyl beta cyclodextrin. So is the sample preparation, and this cyclodextrin can now be also followed easily upon administration. Moving to the other topic, from unlabeled cyclodextrins to labeled cyclodextrins. So the first approach was to use fluorescent labeling. And this approach was used both in vitro samples and in vivo samples, mainly to show how cyclodextrins can penetrate cells, how cyclodextrins can penetrate the surface layers of the skin and stuff like that. Obviously, this starts with a chemical modification. So we put a fluorescent compound and we attach it to the cyclodextrin. It can be done, which is a good thing, with various fluorophores and more or less any kind of cyclodextrin. It's a big change in the characteristics, the physical chemical characteristics of the cyclodextrin, which can lead to changes in pharmacokinetics and pharmacodynamics. So how do the analysis occur? Obviously, it's done with fluorescent microscopy and flow cytometry in this case. And this is a fluorescent FITS labeled HPBeta cyclodextrin. And as you can see, the plasma concentration of the compound, as well as the distribution of the compound and the cell penetration of the compound, can be beautifully followed. And here is another example which is a more complicated, let's say, active ingredient development, but also with fluorescent tagging by our group. And you can also see that it's working very well for visualization. Similar studies were also performed in Japan by the group of Maeda et al., where lactose and hydroxypropyl beta cyclodextrins were labeled in Niemann-Pick C studies to follow the distribution of the compounds after subcutaneous administration. And as you can see, they trace the compounds in different organs, again with fluorescent imaging here. The liver accumulation is shown. So the fluorescent labeling concepts were very well, acknowledging that it will affect the pharmacokinetics of the cyclodextrin and eventually the permeation behavior of the cyclodextrins. Started with the most straightforward way to do pharmacokinetics on cyclodextrins. So as you can see, these results, what I'm showing now, are from the mid-90s, which were even earlier than the first unveil studies that I've shown. From 14 carbon labeled starch, 14 carbon labeled beta cyclodextrin and gamma cyclodextrin were created, and these were already administered in the very first studies. And this is where they showed in the first place that there is no accumulation and of the cyclodextrins in different organs, as you can see, and how they are metabolized or how they are not metabolized and how they are excreted. So this is how the story started, but it's a technique that's currently rarely used. So that's why I decided to put it at the end of the presentation. Still, it's used in some cases, especially when the sensitivity is needed. As I said, it's still an issue with the cyclodextrins. And if we consider how bad cyclodextrins penetrate into certain tissues, this is very important. For instance, in the case of Niemann-Pick C, which is a disease that has both peripheral and central nervous system symptoms, it's very important to see if the cyclodextrins can penetrate the brain so that they can explain the improvement or lack of improvement of neurological symptoms of the disease. So to confirm these, one of the developers of the active ingredient at that time, which was quite recent, as you can see, in just 5 years ago, used, again, 14 carbon labeled hydroxypropyl beta cyclodextrin to confirm if it's penetrating the brain or to what extent is it penetrating the brain. They show that there is no accumulation, and they assessed the efflux rates of the compound with different types of injection. And what they confirmed is that if the cyclodextrin is administered at a fairly low dose, here you can see compared to the usual administration directly into CNS, you can see improvement in the disease symptoms. Whereas if you give a huge dose of the cyclodextrin intravenously, you can see minor to no improvements. Interestingly, what they saw is that the uptake of the cyclodextrins to different parts of the brain, because with this type of labeling this was also possible, is very different. But of course, this type of labeling also made it possible to trace the compound and its pharmacokinetics in different tissues, as you can see here. Not that long ago, in a collaboration with the Debrecen University, we were thinking what could be the alternatives to 14 carbon labeling, because it can be very expensive and very complicated because of the unique reagents that are needed. And if you don't have those reagents, that's very complicated to create these special cyclodextrins. So the idea we had, okay, I made a mistake here. So to not to label with 14 carbon, but with radioactive and I cannot really disclose the details of the chemistry. But let me just say that it's a more general procedure, so you can apply to any kind of cyclodextrin, and it's not that dependent on the reagent. Because of the high similarities between the OH and the fluorine, we didn't see huge dramatic changes in the pharmacokinetics, unlike with the fluorescent tracking. And this compound is prepared in situ technically before the administration. And we used positron emission tomography to trace the material in small animals, in mice. And here you can see the the pharmacokinetics in different organs, as well as a picture of the accumulation, the highest doses. Just as an exotic example, we could say, because it's not really labeling of the cyclodextrin, there's one other radial labeling, we could say a conjugation, that I would like to mention. So again, with some chemical modifications, aminocyclodextrins can be conjugated with different type of complexing agents currently used in diagnostic procedures like NODAGA, and which can encapsulate or complex gadolinium or other radio tracers, and this kind of compounds can again be used to trace the material. Obviously, this modification has a similar impact on the pharmacokinetics of the cyclodextrin, or, well, in principle similar than the fluorescent labeling, so it will change the pharmacokinetics, but these compounds are, again, very well suitable to trace the life cycle of the cyclodextrin within the animals. And some of them can be used actually for diagnostic or therapeutic purposes. So if that's the goal, they can be more useful or ideal. So to wrap up, there are three approaches to analyze cyclodextrins in biological samples. Either unlabeled cyclodextrins are followed, where different types of detection techniques are used currently, but the most modern methods are all LC-MS based. Or you can choose the fluorescent labeling, which is more in the research phase currently, or radial labeling, which is, of course, very accurate for pharmaceutical development, but poses some challenges with cyclodextrins, but it's at least sensitive. In my opinion, what we can conclude is there are now fairly good methods to analyze cyclodextrins in biological samples, although these methods are not sensitive to the extent that would be needed when low-dose cyclodextrins will be used in the therapy. And the labeling of the cyclodextrins still has some room for improvement. So I really hope that this summary was useful for you, and it could bring you some new insights about analysis of cyclodextrins in biological samples. So thank you for your attention.
Thank you very much, Tamas, for your presentation. We are still running a little late, so I propose that we take the question for the panel session, and we go for the next speaker. So let's start now the last but not the least presentation, titled "Promising Future for Hydroxypropyl Beta Cyclodextrin in Inhalation." For this, I'm glad to welcome Dr. Natasha Rox. Natasha first encountered the topic of cyclodextrin during preclinical studies on respiratory models performed in the context of a PhD in biomedical and pharmaceutical sciences at the University of Liège in Belgium. She later pursued experimental work on cyclodextrin used as excipients in formulation intended for treating patients in the field of asthma and lung cancer, before joining the Aculen group in 2019. Currently, Natasha is leading the scientific preclinical and clinical research group of Aculen Pharma, focusing on cyclodextrin-containing liquid and powder formulation intended for inhalation. So, Natasha, the floor is yours.
Thank you very much. Do you hear me?
Yes.
Yes, okay. Do you see my presentation?
Yes, we can.
Okay. So thanks all of you to be here today. I will talk to you today about the promising future for cyclodextrin in inhalation. So what you have to know today is that the current inhaled therapy landscape is an area of high unmet need, but no significant advancement in drug delivery has been achieved over the last 40 years, meaning that the existing inhaled therapies have a very low lung deposition, with only 15 to 30% of the drug that is delivered to the lung there where it is needed. and meaning also that the remaining drug stays in the device, in the mouth, or in the trachea, where it can be responsible for side effects. So improving inhaled drug delivery in respiratory diseases is mandatory, and with this we need to target the organ of interest directly. We need to reduce the potential for side effects and to reduce the number of patients which have an uncontrolled disease, which have risk of exacerbations or which require hospital visits. So inhaled therapies for respiratory diseases such as asthma or chronic obstructive pulmonary diseases strongly needs to be improved. So what are these reasons for this poor lung deposition? So these reasons can be partly attributed to the inhalers that are used right now. So the traditional inhalers that are used are right now the pressurized metered-dose inhalers, which are also called PMDIs. Their efficacy depends on the inhaler technique, meaning that patients need to press the actuation button here on the device to release the drug from the device, and in the same time they have to inhale the drug, which comes out of the device with a very high velocity. So it takes a few milliseconds for the drug to come out. So while the patients are pressing the button, they need to inhale to get the drug. And with this coordination, these patients sometimes have problems with that. And with this, only less than 20% of the drug is really delivered to the lungs. Also, these inhalers are using a propellant gas, which have a negative environmental impact, and this gas will be banned in the future. Then we have the dry powder inhalers. They have a drug in form of a powder. Their efficacy also depends on the technique, and approximately 50% of the patients do not use the inhaler correctly. Because patients, to release the drug from the device, they need to inhale strongly, and this inhalation maneuver is not always performed very correctly by the patient, and which means that they cannot receive the drug correctly in their lungs. And with the current formulations, maximum 30% of the drug is delivered to the lung. Then we have the soft mist inhalers, which are these new inhaler types. These inhalers, they produce aerosols with ultrafine particles, which have a quite low velocity. And here patients do not need this coordination between the actuation of the device and the inhalation, because the aerosol comes out of the device with a quite low velocity. It takes a few seconds for the drug to come out. So the patients, once they press the device, they have the time to inhale smoothly. With this, they improve the drug delivery and they reach a higher lung deposition of approximately 50% compared to the other devices. And these inhalers also are propellant-free, meaning that they have a lower environmental impact. The problem with these devices is that they're not compatible with suspensions, because the aerosol is formed here by pushing the liquid, which is present in the device, through a nozzle which has an ultrafine grid. And these particles which float in the suspensions, at some point they can block the grid, and so they block the device. And so corticosteroid formulations, which are used for treating asthma patients, for example, they are hydrophobic molecules which are insoluble in water. And so these corticosteroid formulations are not compatible with soft mist inhalers. And this is where Aquilon enters the game with the technology platform that could revolutionize inhaled therapies, because Aquilon uses cyclodextrin to complex active ingredients such as corticosteroids. And so in our case, we use mainly the hydroxypropyl beta cyclodextrin, and this complexation of active ingredients with the cyclodextrin allows us to develop liquid and powder drugs which have particular characteristics. So for the liquid drugs, for example, we can complex this hydrophobic treatment, such as these corticosteroids, which render them soluble in water. This allows them not only to increase the pharmacological potential, I will show that in a few seconds, but also to allow the use of these liquid drugs with these last generation inhalers, such as the soft mist inhalers. Also, we do not need to use ethanol to solubilize active ingredients, as it is the case in some commercial references. And also we can sterile filtrated solutions to have a sterile formulation, and with this we do not need to add preservative to our water-based formulation. For the dry powders, we have also formulation technology which allows to produce powders which have particles which have a particular morphology. So we have this golf ball morphology, which confers aerodynamic properties to these drugs. and these particles can fly further and deeper into the lungs. So unfortunately today, due to time restrictions, I will not have time to show you the powder developments of Aquilon, but maybe in a next webinar, we will be able to show you these results. And so our co-founder and pneumologist, Professor Didier Catado, he says that if we want to improve the clinical outcome of the treatment of patients with asthma or COPD, the challenge is to formulate known drugs, so drugs whose efficacy is proven. We need to formulate them in a way that we can achieve a better lung deposition. And so this whole story of Aquilon started back in the 2000s, where the group of Professor Brigitte Evra and her team of the University of Liège in Belgium, they started formulating different cyclodextrin formulations. So they tested the HPBetadex, so hydroxypropyl beta cyclodextrin, the randomly methylated beta cyclodextrin, or the gamma cyclodextrin, and they prepared water-based formulations of these cyclodextrins, and they analyzed the different characteristics of the formulation, such as the density, viscosity, surface tension. We see, for example, that in terms of viscosity, the viscosity of the formulations increase with the increase of cyclodextrin. Also, they evaluated the nebulization rate of these different formulations, which is mandatory if you want to aerosolize your formulation for inhalation administration. And they saw that the nebulization rate decreases with the increase in concentration of cyclodextrin. And there was even a level below which you should not go of 0.2 gram per minute, because below these formulations, the nebulization of these formulations are not compatible with a human administration, because it would take too long to nebulize the formulation. This allowed also to fix the maximal concentration of cyclodextrin that could be used in these liquid formulations. The university also performed an in vivo assessment of different formulations. So here they tested the formulations at different concentrations, and they administered for 7 consecutive days these formulation by inhalation administration to mice. And so these aerosols were produced by using an ultrasonic nebulizer, and the animals were exposed to these formulations for 30 minutes per day. At the end of the experiment, there was the animals were sacrificed, a bronchoalveolar lavage was performed in order to investigate the number and the type of inflammatory cells that were present in the lungs. So they assessed the number of neutrophils, of eosinophils and macrophages, and we did not see any event that was suggesting an inflammation in the lungs. They also analyzed the lung tissues under the microscope. They assessed for epithelial integrity or peribronchial inflammation. We saw that the epithelial integrity was okay. There was no peribronchial inflammation. The kidney tissues were also analyzed, and they looked perfectly normal. The lung tissue, the lung function was also assessed via plethysmography, and the lung function was okay. So this suggested a safe pulmonary administration of different types of cyclodextrins that was performed by this University of Liège. And so, knowing that we had this safe profile of the cyclodextrin, and also based on what was published in the literature on the hydroxypropyl beta cyclodextrin, we saw that this HPBetadex had a very safe profile, also by other rules of administration, and that's why the university started trying to solubilize budesonide by the cyclodextrin. So budesonide is an anti-inflammatory molecule which is used widely to treat asthma patients, and this complexation of the budesonide with the cyclodextrin worked, and so the university managed to solubilize the budesonide in water, and these complexes that were formed had a very low cytotoxicity. Budesonide had its anti-inflammatory properties, which were preserved. I will show that in a few seconds. And we also saw that the complex formulation had a better deposition, and in vitro assessment of a lung deposition, this deposition was increased in vitro. And so these complexes were developed for the indication of asthma, of COPD, or of allergic rhinitis, for example. And so they performed at the university proof of concept of the budesonide cyclodextrin complexes in a mouse model of asthma. In this mouse model of asthma, they were using ovalbumin. Ovalbumin is a very well-known allergen that is used as an allergen to mimic the asthma characteristics in mice. So mice which are challenged with ovalbumin develop a pulmonary inflammation, which is mainly composed of eosinophils. And so these mice here were sensitized with ovalbumin, and then they were challenged during 5 consecutive days with ovalbumin aerosols. And before the ovalbumin aerosols, these mice were treated intranasally, either with the cyclodextrin alone or with the budesonide alone in form of a suspension. So it was a liquid formulation in form of a suspension. So it was the commercial reference Pulmicort that was used in this experiment, or we used the budesonide cyclodextrin complexes. At the end of the experiment, the animals were sacrificed and the lungs were collected and assessed for peribronchial inflammation under the microscope. So here, concerning this peribronchial inflammation, it was analyzed, and we see, for example, that for mice which were not challenged with ovalbumin, we see that they had very mild bronchi with no much inflammation around, and so the peribronchial inflammation was quite low. For mice which were challenged with ovalbumin here, and which were treated with a control buffer, we see that we have inflammation around this bronchi, and the score is higher. Same for mice which were challenged with ovalbumin and treated with the cyclodextrin. Mice which were challenged with ovalbumin and treated with the Pulmicort, here we see that they still have a peribronchial inflammation, and this was the dose of 100 microgram ml of budesonide. Now, when we use the budesonide suspension at a dose of 250 microgram ml of budesonide, we see that the inflammation score is reduced. Same for mice treated, challenged with ovalbumin and treated with the complex at a dose of 100 microgram ml. So we see here that we have a similar efficacy between Pulmicort and the budesonide complex, but at a 2.5 times less budesonide dose. So this was quite motivating, and the university then started the budesonide permeability test across a cell layer. For this, they used Calvi-3 cells, which are lung epithelial cells, that they plated in a transwell. And so these cells were left here to culture for several days. These allowed the cells to form tight junctions, to develop a mucus layer above the cells. This was really mimicking the conditions in the lungs. And then on top, they plated formulations of budesonide and cyclodextrin with increasing cyclodextrin doses. The cells were left like this to incubate for 4 hours, and after these 4 hours, they collected the medium present on the apical and on the basal side, and they quantified for budesonide levels in this medium. And this allowed to assess the budesonide passage speed in function of the concentration of cyclodextrin. And we see that with increasing doses of cyclodextrin, we have the budesonide passage across the cell layer, which was reduced. We see this also here in a way where we test the permeability, so it's another way to present the results. So budesonide alone has a high permeability. When we add to this budesonide cyclodextrin, we see that this permeability is reduced. When we now keep the same level of cyclodextrin and we add the budesonide in excess, we see that the permeability of budesonide increases again. So this suggests that the flux or the permeability flux is related to the free budesonide fraction, suggesting that the cyclodextrin acts as a sustained delivery system or as a reservoir which slowly releases the budesonide from the complex, and which suggests a prolonged activity of the budesonide in the airways. So after that, we at Agrilon, we started the toxicology program of these complex formulations by inhalation administration. This was mandatory because we need to somehow qualify the cyclodextrin when administered by inhalation, and we needed to de-risk our development. And so for this, we performed 2 studies in rats, a 2-week dose range finding study and a 4-week GMP study, where the complex formulations were administered during 2 or 4 consecutive weeks to rats. We also started a study in dogs, which was a 2-week exposure study. So for the animals which were exposed to the budesonide cyclodextrin complex, basically we saw expected class effects following the administration of budesonide. These effects are very well described already for the budesonide. What we also saw is that our formulations were respirable to the lung. This seems not so important, but it is. If you want to administer by inhalation, you have to have droplets which are compatible with the lung deposition. We did not see any unexpected toxicology finding upon HPBetadex administered by inhalation, and these toxicology studies allowed us to start our clinical program. And so we performed a single dose study in human healthy volunteers and a 4-week exposure study in asthmatic patients. So concerning our clinical program, so I will show you here the results of this first human study of the complex formulations, which is called Boreas. It's a phase 1/2a clinical study that was performed in 24 adults with mild asthma. So these adults were receiving in a random order either the budesonide cyclodextrin formulation at a low dose of budesonide, or the budesonide teva, which is the commercial reference, which was the comparator, and it is a budesonide suspension. So patients were receiving the formulations for 28 consecutive days, and after a washout period of 14 days, patients were receiving the other treatment, so it was the other way around. These formulations were administered here in our case with the Pariboy Classic nebulizer, and the formulations were administered once per day. So first of all, of course, we assessed for the safety, and we were lucky and we did not show any adverse event which was related to the product. This was already excellent news. And also we assessed for the efficacy. And for this, we performed a PC20 metacholine challenge test. This PC20 test was performed in the very beginning, so before any treatment administration, and then it was performed at the end of each treatment period. PC20 is the concentration of metacholine that induces a fall of FEV1, so a forced expiratory volume in the first second of 20%. Meaning that patients were at each PC20 testing, patients were receiving increasing doses of metacholine. Metacholine is a bronchoconstrictor, which will make your lungs getting constricted more and more, depending on the dose of metacholine that you receive, and patients will also have problems in exhaling very strongly. And after each metacholine inhalation, patients had to perform a spirometry test for which they had to breathe or exhale very strongly in an equipment. And this equipment was measuring the volume of air that was expired in the first second. Once this volume of air, which was expired in the first second, decreased by 20%, we stopped the test and we measured the levels of metacholine that induced this FEV1 fall of 20%. Meaning that the higher you can go with the concentration of metacholine, the better the patients feel and the less hyperresponsiveness they have. So what we saw here for patients after the treatment with the budesonide cyclodextrin complex, we see that we could go higher in the concentrations of metacholine when compared to patients which had been treated by the budesonide suspension. And so this means that after the budesonide cyclodextrin treatment, patients had better asthma control and less hyperresponsiveness when compared to their treatment with the budesonide suspension. So all in all, we can say that Aquilon's HPBetadex-based formulation, which are administered by inhalation, but they are respirable, they are safe. We showed that with animal and human data. We reached the proof of concept as these formulations of budesonide cyclodextrin provide a better asthma control. We saw that in animal and human data. And this opens the way for future developments where we can combine our formulations, including these corticosteroids, which are insoluble in water. We can combine them with last generation inhalers, such as these soft mist inhalers. This will not only improve the lung deposition, because we will use last generation inhalers, but we can also improve the pharmacology of the active ingredients. Our developments are patented. And we see a very bright future for inhalation therapies because we can complex other non or low soluble molecules which would require topical lung application, for example, PDE4 inhibitors, monoclonal antibodies. Also, the spray drying of biologics with HPBetadex will prevent these biologics from shear or thermal stresses. We can also prevent aggregation post-drying, and also these liquid protein formulations which use HPBetadex can have an increased stability, and also we can reduce the absorption of the proteins on surfaces, etc., etc. So thank you very much for your attention, and happy to read your questions.
Thank you very much, Natasha, for the very nice presentation. So I suggest that we open now the panel session. And for this, I am glad to welcome Paul Maes, who is Chief Executive Officer of Aculent Pharma, and 3 colleagues from Roquette, experts in cyclodextrin, Julien Park, who manages the functionalization laboratory for starches, sugars, and polyol, Dr. Damien Truffin, who is pharmacologist and toxicologist, and Hélène Blouet, who is head of biopharma technology at Roquette. So maybe we could stay in the inhalation field with the first question for you, Natasha. So Natasha, based on the first in-human study that you have done, showing no adverse effect, do you foresee that hydroxypropyl beta-cyclodextrin can have or do not have effect on the lung when inhalated?
Yeah, basically, based on our studies, we showed, thanks to the animal studies where we used increased, so very high doses of cyclodextrin over sometimes over 2 or 4 weeks. So we went until 160 times the clinical dose. We did not see any effects on the lungs of the animals. Also, indeed, as you say, the clinical results are very reassuring based on what we saw in the patients. And so I would say that at the doses that we will use of cyclodextrins, we will basically not see any effect. But of course, I cannot promise anything. We need, of course, to go further in the tox assessment. We need a longer time of administration. But at the doses of cyclodextrin that we test, I don't really think that there will be any effects.
Okay, thank you, Natasha. One question for you, Paul. How do you see the future of inhalation route of administration?
Well, that's an area which has not been really explored. With cyclodextrin, as Natasha showed, you obtain a pseudo solution with some reservoir activity, and therefore many things can be done. We could see that the pharmacology of ICS is changed because of the presentation of the active to the lipid layer, lipid rafts, as we could see in the video. But other drugs will not be changed very much. So each time, as said before, it's a new area to test, both in vitro, ex vivo, and in vivo. But I see more and more a huge potential in biologics, because biologics, you don't want to send them elsewhere than where they need to act. And with the technology using cyclodextrin, you can reach not yet attained amounts of drug into the deep lung. Up to 80%, we believe, is feasible.
Okay, thank you, Paul. Maybe coming back to Tamash's presentation, Tamash, where are the methodology gaps we have to fill in the future?
Well, that's a tricky question. So I think as I said during the presentation, I believe that the sensitivity will be a very focal point in the future. Maybe that's not a methodology problem to solve, because HPLC MSMS seems to be a reliable technique to detect cyclodextrins. But as I expect, the doses will get lower in time, especially for applications where they are used as therapeutics, and for that we need to either use a better sample preparation techniques to have higher sensitivity, or to improve the analytical tools themselves. I also see potential in using radio labeling other than the carbon, as I mentioned, to be something more convenient due to the lack of radioactive reagents to make for carbon labeled cyclodextrins.
Okay, thank you, Tamas. Another question, I mean, for Balázs, since intravenous and oral are the most used application, what emerging non-traditional formulation will dominate the future application, according to you?
It's a very good question, and I can tell you my opinion, but I think it's just an opinion. But I'm really sorry that I didn't put a graph in my presentation where we could show that that patents and technologies are emerging, but in the same trends. So always the IV solution and oral administration is leading, and these trends are staying the same. So it's just an opinion, but I think that the nasal administration and the pulmonary administration are the most promising, I think, based on the articles and based on the patents I see nowadays.
Okay, thank you, Balázs. Thank you. Maybe now a question for you, Carmen. Among oral formulation, which types are dominant, and are cyclodextrin compatible with all kind of oral formulation?
Of course, compatibility depends on how do you want to prepare this formulation. So I just show some few methods for doing solid dispersions for solid dosage form, but there are many other options. Always depends on what you as a company have in-house, because these are not cheap equipment. So you have to go with very, very successful. Let's say besides tablets, you can go with oral disintegrating films, oral disintegrating tablets. You can create emulsions. You can create a lot of oral dosage form. So I think just the imagination is the limit.
Thank you, Carmen. Going back to inhalation, maybe a question for you, Damien. What are the special safety consideration for the inhalation route?
Thank you. In fact, for safety concern, there is no big concern for cyclodextrin. But of course, we always have to consider the dosage. And if we consider very high dosage, we have to be sure that there is no absorption of cyclodextrin into the bloodstream, because we saw that at very high dosage, we could have a passage to the cell layer. So it's very dependent of the concentration. And another point, of course, like other excipients for pulmonary routes, nasal routes, and all the mucosa contact, we could have irritation effect at very high dose due to cholesterol on trapping and cell lysis, who could lead to irritation and so on. But this phenomenon occur at very high dosage. But always we have to check this point, I think.
Okay, thank you, Damien. Maybe a question for you, Paul. How does the hydroxypropyl cyclodextrin budesonide compare with the captizol-enabled budesonide published human use?
There are two major differences between the two. The constant for exchange with the HPBCD is quite high, and the amount needed to create a reservoir effect like the HPBCD is a bit high. On the other hand, it has an effect on osmolarity because of the ionic nature, and maybe you know, but in inhalation toxicology, there is a factor 10 for rat toxicology. And if you already need a safety factor of 50 plus 10 times, then you achieve major osmotic pressure. While the HPBCD is non-ionic, so the osmotic pressure is low.
Okay, thank you.
On the other hand, we have data showing that using the reservoir effect, you don't change much to AUC and Cmax. You change it a bit. You change more the activity than the pharmacokinetic parameters
Of the activity.
yeah. Of the ICS.
Okay. Carmen, you want
Yes,
to say something?
No.
Ella, sorry.
Yeah, if you don't mind, Nicolas,
Yeah.
I'm jumping in. Instead of answering question, I would like to address, let's say, a question to Natasha and Tamas. Actually, Tamas made us a nice overview of the analytical method in biological product, but Natasha, she experienced this. She made it on rat, dog, animal. So I have this question for both of you. Let's start with the one who made the review and then the one who experienced it. What are the main challenge? Actually, what is the main challenge when you do testing? Because this is coming very often, asking, how can I test my HPBCD in biological product? So what are the main challenge that you can foresee? So depending on, we give the floor to Natasha first, since she experienced, Tamas. Okay.
Yeah, thank you, Elan, for this question. So let's take like this. I'm first of all not an expert in bioanalysis and in analytical methods. So for me, all these methods are already quite complicated. And while Aquilon was developing these methods for investigating the dosing of cyclodextrin in fluids, I was not there yet. It was my colleague Marie-Astrid Albert. She was developing these methods with a partner at the University of Liège. And I remember that it was extremely complicated in the beginning by the time to find the right equipment, the right methods, parameters, and to have enough sensitivity to be able to find the methods. Now, from my experience, now that everything is running, basically, I think that we perform quite fine on our methods, which are done more in-house, but also with from the toxicology samples, we have enough sensitivity, and we can have quite nice dosages. But I remember that the setting up of everything was very complicated, and that it was quite stressful for her. I remember this very well because also for inhalation, also not only in the biological fluids, but also when you need to characterize your formulations so that they are compatible with the right inhalation at the CRO that are performing the toxicology assessments, you need also to develop a method. And she was also struggling with these partners because you really need the right machine and the right sensitivity. So, voilà, that's basically what I can say. I don't know, Paul, if you have...
Natasha. Tamas?
Well, in this particular case, what Natasha mentioned, I think that would be the key here. So when you administer orally or parenterally a cyclodextrin-based formulations, usually there is a lot of cyclodextrin included. For an inhalation formulation, inherently the dose of the cyclodextrin is much lower, so sensitivity will become very critical, and especially from that essentially low amount of cyclodextrin, even less will be in the systemic circulation. What I guess here the additional challenge was that they are, the pioneers in pulmonary administration, so there was no available data to use or to compare, but Paul can confirm it if what we could rely on to show how is it, for instance, accumulating in pulmonary tissues.
We needed also to develop the method for the matrix of pulmonary tissues, indeed, of mice, of dogs, or whatever. So this was always also a challenge to have this species.
Paul, you want to add something, Paul, please?
Let's say we basically use the best labs with the best equipment in CMSMS. There is no other way so far for what we do. you just have to prepare the big check for the analytical lab, the experts.
Okay, thank you so much.
Thank you very much. I think we are reaching the end of this webinar. So thank you very much again to all the attendees and the speakers, and special thanks to Paul. I remind you that you will have online access to the recording of this webinar within the next days. So I thank you very much again for your attention, and bye-bye. Have a good day.
Bye-bye.
Bye.